Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of the discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
So I was looking at our case studies, and I think today's might actually be the simplest one.
Which is next-gen sequencing?
Yeah, because that's very clearly a test and most likely going to be high risk. In my opinion, I guess I'm eager to hear what others think.
I agree. It's not like a companion or a diagnostic aid, right? It gives you a discrete result.
Yep.
Yep. So let me just take the slide deck and then I add our notes here. I think this is the current version. 1 second. All right. So next generation. So we're probably talking about a tumor panel, correct?
Most likely.
Yep. So this is not a companion. And, you know, I don't know. Should we discuss sort of the intended use? I mean, of course, you know, treatment stratification or whatever we want to call it, right? Okay, so, you know, following sort of the outline that we have, sort of, you know, there could be irreversible harm done. Of course, you know, some panels can be used for diagnostic purposes, right? If, you know, a key, whatever fusion or key mutation is identified, it may lead to reclassification of a tumor, which I would call that high risk as well. Now, just briefly to discuss that, do we think that there are mitigating measures to alleviate that risk, or are we just saying definitely not possible?
Well, I mean, I think the thing is that, you know, if you're just reporting a discrete result, so if you're just saying, this is lung cancer, I'm only testing for EGFR, it's EGFR negative, I think that's pretty high risk because there's no additional piece of it, like if that's an end-to-end solution. If on the flip side you have, you know, this is where the mitigating measures get a little complicated, right? Because there's going to be assay mitigating measures, so like quote unquote special controls and things like that, which I still don't really understand completely. But And then there's the professional piece, right? So if you have a pathologist who, is trained in anatomic pathology, they're looking at the tissue, they're saying, yes, there's neoplasm here, they're selecting the area, it's going through the thing. And at the end of the day, you get it's negative for EGFR, but it's positive for KRAS, right? And you can say, okay, based on my understanding of performance and my understanding of pathology, this is a KRAS mutated lung cancer. It likely doesn't have EGFR. I think that it's a mitigating measure, right? Because you're applying your professional judgment in the case. And so, and also, similarly, if I look at it and I say, well, it was a borderline sample to begin with, I'm not identifying anything, there's no other mutations, there's no TP53, there's no other drivers, I'm not confident that this is a true result. And I qualify it as EGFR negative, but sample quality isn't adequate, recommend resubmitting a new sample, right? I think that's a qualified negative result, and the risk is lower.
Yeah, I think of special controls a lot of times as like quality assurance activities. We have well-trained staff, we've got quality controls that we run, a whole host of things that we do in the labs already. I think a lot of those would fall into kind of special controls. The other piece that I think could potentially be a mitigating measure is how well understood the gene in question is. So FDA has kind of this stratification. I don't know if it's FDA, but there is a stratification anyway of genes. Like, we know that this gene is associated with this disorder. There's evidence this gene is associated with this disorder, or there's incidental information. So it's kind of degrees of certainty with how well a particular gene is associated with the disease. I think that could probably be a mitigating measure as well.
So this goes more into, the AV/CV, right? Like clinical validity is, what is you mentioned like what's the prevalence in a certain disease, right? So versus like some of the other elements are more probably affecting the analytical validity of the test.
I think that's fair.
So, special controls, would we consider run controls? Let's say, what New York State says, like let's say positive, negative control on the same run, or would we consider that special controls, or?
I think so. And those of you who have more experience with the FDA certainly chime in, but I think special controls really are for this particular test or this particular methodology or type of test. What are the special things you do to make sure that you're getting accurate, consistent results? Is how I interpret special controls.
My general thought on this is it seems like it's almost always some kind of physical thing. So like you said, like it can like a additional step, additional control, material, whatever. But as best I can tell, and in discussions with folks at the FDA and elsewhere, they never really answer the question whether or not the expertise of a person looking at this qualifies as a special control.
Yeah.
So, and I think that's where it gets a little challenging. And so my pet example is, cell-free DNA, right? So cell-free DNA, we can find a lot of these same mutations in people with different cancers. They're used for treatment, et cetera. And so until a couple of years ago, occasionally you would see TP53 mutations, you see DNMT3A mutations, you see TET2 mutations. And the way that a lot of the industry was interpreting that is, oh, there's clearly a neoplastic process there. This is related to your cancer. Therefore, you don't have the EGFR mutation we'd expect in this lung cancer. And the problem is that they were totally, the way that those reports are written is that they weren't considering clonal hematopoiesis. And so, that is, yeah, you could program something in your pipeline to say this is, potentially clonal hematopoiesis, but it's hard to differentiate from a subpopulation. But at the same time, it's easy to say in a person, hey, this TET2 mutation is more associated with a hematopoietic process, and exclude that. So we had patients who, we had seen who were kept on chemotherapy when they had a targeted option based on an incorrect interpretation of an analytically valid result because there wasn't that professional and experiential piece included.
So this seems to be similar to more like there's two competing diseases now where, I mean, this gets to the almost to the point of like histology, right? Where, I mean, you take the biopsy to check whether it's a metastatic HER2 breast cancer, and what you find is actually a nodular liver process that's something completely different. So it's two diseases presenting as one, and then you try to actually work up, in this case, you know, in Eric, in your lung cancer, you work up to see whether you find a TP53 mutation in lung cancer, but you actually identify clonal hematopoiesis with a tattoo or something like that. So I would consider that sort of like in that CV bin, but it's very hard to distinguish both, right?
Well, that's where, you know, you need that experiential piece, and you need that judgment piece. And I think that's really hard to, you can systematize it to some degree, like, oh, okay, you should always think of, you know, my very first day on AP as a resident, my attending said that, you know, when you have something that's poorly differentiated, you know, you think about epithelia, you think about mesenchymal, about hematopoietic, and the very last thing you do before you do anything else is you hit yourself on the leg and say, could it be melanoma? And so that particular person would get, you know, a melanoma stain on every single poorly differentiated cancer. And sometimes when it wasn't poorly differentiated, yeah, it was systematic. It wasn't necessarily the best way to do it, and occasionally went down the tubes because of it, you know. So, you know, is that, you know, there's using something like that versus using, you know, judgment and expertise to select when you're going to do something, right? And so then there's competing needs in terms of like how you're, you know, costing the healthcare system and your patient and giving diagnoses that may or may not be entirely perfect, versus, you know, making sure you never ever miss something. So again, it comes back to, I think there are controls you can use. And so I think of, you know, if you know your assay, so I think about assays I've developed, and, you know, I know my assays, a hotspot amplicon assay was very susceptible to indels of a particular length. And once you got to a certain length, then the sensitivity went down. So my routine control was at, you know, just above where the assay performance started to have problems. And it was an indel. And so anytime there was a problem, that control would basically go out of range, and then we'd know there was a problem. So I consider that a special control to kind of help assess for something that's a known problem.
Yeah, so I find that very interesting. So for example, like, you know, separate cutoffs. I mean, we discussed that with our assay, separate cutoffs for point mutations versus, as you mentioned, indels, right? I mean, clearly indel callers are much more intricate and harder to do, you know, 5', 3' alignment, all these intricate points. While point mutations, I think the callers are just more straightforward. You can even do compound callers or, you know, use multiple. I wanted to come back to something with the melanoma example. So I think there's an implied claim with an H&E exam that when we say there's no evidence of malignancy, you know, in the colon, that means, you know, we excluded colorectal cancer, right? It's like context dependent. But if you have like, you know, some like weird wound, I mean, it's implying that the examination also excluded all other kinds of tumors, lymphoma, melanoma, right? But that's never explicitly stated. And I think that reminds me of, you know, I mean, some people would really like to have the pertinent negatives on an NGS report, right? Did you check KRAS hotspots and BRAF and, you know? So, I mean, that probably comes in FDA terms to the claim, right? What are you claiming to provide with this test? But I mean, I think we're probably okay with, like, to go back to this sort of thing. Is it low risk, high risk with mitigating measures, or high risk? I think we probably all agree that this, like, NGS tumor panels are probably going to be high risk, right?
Yeah.
So what would we consider then, you know, because we probably would need to demonstrate at least in some way, shapes, or form the clinical validity, correct? Under VALID?
Yes.
So, I mean, we can, you know, take a simple example, right? We have whatever, EGFR, ALK, BRAF, or something like the common targets. because you can probably, or I mean, at least that would be the assumption, we can start with those, let's say, 3 that are rather common. I think that's what Foundation Medicine also did, right? Like HER2 amplification was also assessed, and then demonstrate clinical validity on some 4 or 5 key targets, and then sort of submit that. Could we briefly discuss what that would look like for a laboratory? You know, because like clinical validity demonstration for a laboratory can be tricky, right? Or
You can always do a clinical trial, which everyone's really excited about.
I was hoping we'd talk about this tiny piece of work. I mean, that would be then, of course, the whole landscape of, you know, the whole trial landscape of, you know, I don't know if you saw this recently. Let me just trying to pull this up. There was a beautiful Cell article about the landscape of targeted trials. I don't know if you saw that. Let me just quickly find this. Here it's this piece here. Let me just see if I can rotate this. So this was a snapshot of trial types in precision medicine. So if we are being asked to help design trials, there's, you know, of course, interventional, there's real world data, so we can zoom in on that briefly, and then of course master interventional trials, of which, you know, Basket and Umbrella is probably the most well known. And then there's these master observational trials with like very broad inclusion criteria. But, you know, when talking about the real world data, which I think is probably most helpful to many labs, there is a retrospective observational trial. So basically, you know, chart review or medical record review to see whether, you know, patients had the same response as, you know, in interventional or randomized control trials. So that is, I think that's the sort of connotation here, that in the first bucket, there's, you know, of course, many types of trials, right?
Yeah, so in the definitional section of VALID right up front, they have a definition for valid scientific evidence, and then they have a laundry list of things that can be used to demonstrate valid scientific evidence. So I think those are all elements that a lab could consider to sort of build the case that you have a clinically validated test. So, you know, peer-reviewed literature, bench studies, case histories. They talk about real-world data. A big one is data registries for NGS. You know, FDA does have a process for recognizing data registries. So the good news is, I think labs will have a lot of options beyond doing, you know, an old-fashioned clinical trial.
Do you mind like giving me like a few of those so I can just add that to the slide deck here?
Sure. So peer-reviewed literature. I think that's kind of what we really lean on today. Case review, data registries.
And what does case review mean?
That'd be like a case history review. So like, you know, I have a patient who I know has XYZ cancer, you know, just due to their clinical picture, and I can demonstrate that my test is positive. My test for that cancer is positive. That's a really terrible example, but that's the gist.
So it's at the individual patient level, basically.
Correct. Yep.
Okay. So this would likely be probably, I mean, I just want to make sure that we have, so orphan disease would probably fall in that group, right? If you have like an extremely rare event or so. I know that there's an orphan disease exemption, but you know, like, I don't know, I'm just trying to figure out like where would an individual patient level matter the most.
Well, I think where this really comes up is in academic medical centers, right? Where, you know, you're seeing patients a lot of times, you know, for more rare or esoteric, you know, disease states, and now your AMC wants to develop their own lab developed test or IVCT to help out your clinical practice, and so you partner with your physicians in the clinical practice to get, you know, case histories on these patients and probably samples as well as you work to develop your assay.
Okay. So this would be like a, like specifically developed LDT, sort of. right?
Yes.
Okay. Okay, then data registries, I think you mentioned, right?
Yep. And then, of course, like you'd mentioned, real world data, real world evidence.
Mm-hmm.
Let's see some of the good ones. Post-market data. So I suppose that might be relevant if, someone else has a similar test on the market, but for whatever reason you want to develop your own, you potentially could leverage data from that other test, maybe.
I mean, that would probably go to like, what's the prevalence in certain diseases? I mean, that would be, yep, other.
I know some groups have floated the idea of some sort of a conditional approval. Like if I don't have enough data to, prospectively demonstrate, really strong clinical validity, could FDA give me conditional approval? I bring my test to market, and then I collect that clinical validity data while the test is, being performed on patients, and then 6 months later, a year later, whatever, go back to the FDA and provide that clinical validity data at that time. Now, I don't know if that's going to fly with the agency, but that's another kind of use case for that post-market data idea.
I mean, that would be almost like, then if this is the designated test, right, the what is it called, the pre-certified assay, you would have to report that back anyway, right? So that could be tied to that sort of rule. Yeah, interesting. So this part here, I think the case review would be just at an individual level, the same as real world data and real world evidence, but just at a very specific, probably finely granular part. The data registries, I think you mentioned, so some data registries are FDA approved. So I presume some people could say, look, we mined ClinVar and we found the same rate of, somatic mutations, and, since ClinVar is FDA approved or authorized, then that would make sense. I think the peer-reviewed literature I find interesting because, at least when we're giving lectures to our trainees and we talk about what a validation looks like, it's basically what any credible, peer review process would ask you for, establishing the performance of a test, right? You would ask for reproducibility and sensitivity, etc. So I think that's a very interesting approach. So I don't know if you heard, but in the jurisdiction that Massachusetts is in, there was just a proposal by National Government Services that would say that next-gen sequencing assays, if they have either FDA authorization or peer-reviewed publication, they would cover it at the increased rate. But it's still a proposal, so there was just a hearing about it, but clearly this idea of peer-reviewed literature acting as a surrogate for increased quality is implied there. So I can see if I can check that proposed local coverage determination, what the exact wording is. So maybe I can paste it here. But that part I think is really an interesting approach because, for most AMCs that creates at least a mini incentive to, write up the validation. I think a lot of people do that anyway, but if that would be another option to demonstrate that would be great. Did they mention
up to this point, FDA has been pretty clear that peer-reviewed literature alone isn't enough to demonstrate clinical validity. I'm a little curious to see if they soften that approach with IVCTs, or if you can use peer-reviewed literature, but it needs to be in conjunction with one of these other options.
Mm-hmm. Is anyone aware, is New York State allowing peer-reviewed literature?
Yes.
Yes. So then you don't have to submit all your data, or how do they handle it?
Well, no, you leverage peer-reviewed data as evidence of clinical validity. So, I have these 3 papers that say this biomarker is associated with this disease. We have developed a test for that biomarker. Therefore, our test can diagnose that disease.
But you would still, I mean, we only have 2 tests submitted, so I'm not too familiar with the process, but you would still need to demonstrate the whole analytical validity piece, irrespective of whether that paper came from you, right?
Correct.
So it's like, so New York State does not accept peer-reviewed literature as an equivalent
To analytical validity, no, they do not.
Yeah. So I mean, this is vastly oversimplified, but
yeah, I mean, the peer-reviewed literature is really addressing the clinical validity question. You still have to demonstrate, the full analytical validity.
Yeah, so I thought that for NGS that's a particular interesting thing because with the other examples that we had Gleason grading, etc. For this step of saying, so analytical validity is, I think most labs would say yes. If you're willing to take the risk with an LDT, you probably are competent and feel at least that you can do the analytical validity. The clinical validity is somewhat more tricky, but New York State, you can basically use what others have demonstrated as a surrogate for that. And then by demonstrating, hey, here's the 6 New England Journal papers on BRAF, here's our analytical validity data. That's how they bridge, so that you as a lab do not have to demonstrate in a trial that BRAF detected works. So at least that's how I understand it. But that part I haven't seen how that would work under VALID, right? Because on the one hand, there's the expectation that you really demonstrate clinical validity, but whether the peer review could substitute for, let's say, local in the laboratory or in your setting clinical validity, or whether you could use, I mean, does it state that peer-reviewed literature would be sufficient?
So it basically says you need to demonstrate valid scientific evidence of clinical validity. Valid scientific evidence is defined as literature, case review, data registries, etc.
Got it.
And then just my one bit of commentary is based on previous experience, granted under the current, medical device framework. FDA has shared with us that peer-reviewed literature alone is not enough to satisfy clinical validity for them. You'd need something above and beyond peer-reviewed literature. So it's unclear at this point in time if, under VALID, if I submit my NGS test and the only clinical validity I have is some papers that show that the genes I'm targeting are associated with XYZ cancer, is that sufficient? Or will I also need one of these other elements of valid scientific evidence in addition to make the case that my test is clinically valid?
I mean,
it's interesting too, if you read through all the parts in that part B there, I mean, it is peer-reviewed literature, but also clinical guidelines, bench studies.
Yes, there's a lot more.
Yeah, and I mean, the clinical guidelines is kind of interesting. So, which clinical guidelines, are NCCN guidelines held up, higher than somebody else? And then clinical trials is kind of one of them, and then also data collected outside the US.
And then comes the beautiful word best practices, right? Because you can say, like, it's best practices to do TMB, PD-L1, MSI, and everything, but, whether there's any good data and evidence that you need to do all for all, that's like a completely different topic. But
yeah, so I think FDA usually, they have a process to get standards recognized, FDA recognized, meaning then you could use them. So like, for example, CLSI, broadly speaking, is an FDA recognized standards organization. So if, as a lab, I want to write an accuracy study, if I'm going to the FDA, they're going to expect me to follow the CLSI guidance document on accuracy because it's a recognized standard. So whether it's, one of these other professional organizations you mentioned, if they get, if one of their standards get recognized for, here's how you diagnose XYZ cancer, that could likely then be used as valid scientific evidence for clinical validity.
no, it's a good point. So, I mean, there is the FDA database on recognized standards, so it's probably like shuffling it together. I mean, it goes a little bit beyond NGS, but I know the JMD and AMP work on, best practice and guidelines on how to validate NGS assays. I presume that the FDA would recognize that if you follow those point by point, that you do a better job than if you don't. I mean, it's kind of an interesting thing.
And I have no idea how easy or difficult it is to get a standard recognized by the FDA. I don't know if Eric or Rajni or Joe, if you have any experience with that.
So now at some point we spoke about, with respect to whole slide imaging, we spoke to the FDA about DICOM, because DICOM has been and is a recognized standard, but there hasn't been any submission in the whole slide imaging field made using DICOM. So there's also, even if the standard is recognized, the recognition of the standard follows usually at least one product being on the market using that standard. So that's a very bizarre conundrum, because in mammography and in other radiology applications, the standard is implemented and accepted and used, and then the FDA recognizes it. But with DICOM in whole slide imaging, it's not there because it's not used. But if it would be used, they would recognize it. It's kind of like, I mean, that's as far as my experience goes on that part. But
Interesting.
it's like, I think sooner or later someone will identify the power of DICOM. It's just a lot more coding and a lot more, I mean, right now SVS files are like sufficient, right, for many applications. But the converters, the de-identification, and all those tools are amenable. So I think it's just a matter of time.
Yeah, I mean, demonstrating clinical validity is definitely one of the kind of higher priority things that we're concerned about, at least, because that can be very time consuming and expensive depending on what you need to do. So I do think if VALID goes through, one significant service that professional organizations can give to their members is really to develop standards and get recognized by FDA.
I mean, the part on, of course, the additional cost of, I mean, the FDA would probably like if there's a trial run, right? If you have a trial and have that data and demonstrate with a certain assay, that would be great. But then you have to somehow fold that cost back into your individual cost and or some other way to cover for it. So that creates a huge disincentive for smaller labs to build their own assay, which I get, but then that would mean it creates absolute incentive to work with commercial products. But for rarer diseases or for local expertise, that I mean, I don't know whether you can justify everything with the rare disease exemption, but the local development, if that's if the high risk goes along with demonstrating CV, like this whole thing starts sort of an avalanche of consequences, right? To say the additional cost of demonstrating that, I mean, it's, yeah. What do you guys think? I mean, for an NGS assay, right? The demonstration for like 3-4 targets is, I think, manageable, right? With chart review and looking at it. But that's still, you have to probably demonstrate, is this patient population representative of, like, if we pull 20 lung cancers, is this representative of our local lung cancer population, and is our lung cancer population representative of lung cancer? And I know that those questions is always like extremely relevant to the FDA, and of course we're talking gender bias, right?
It's a hot topic right now.
Yeah, and so I mean, how would I mean, I would say even with hundreds of patients that we test every month, that is still a tough topic, right? You may not even get patients on drugs because of various reasons, right? Like too sick, financial assistance programs are not working. So even if you analytically identify a subset, it's almost like an inability to demonstrate that.
Yeah, it's a challenge. I mean, in Minnesota, if we're developing tests for a bunch of white Norwegians, we're great. But, if we're doing other patient populations, it gets a little more challenging. So I think I agree, Joe. I think that's going to be a significant challenge for laboratories, is getting samples for validation and cases for validation that is representative of the patient population.
Yeah, so I mean, ability to demonstrate representativeness, is that a word? Sorry.
It is now.
I just mean that the data set is representative.
So, but the data set has to be representative of the samples coming into the laboratory because we're talking about an LDT. So it's just for that specific lab, right?
Yes.
Yep, that was too much in one word. Sorry.
But I think this is compounded by the fact, and I think we touched on this, is due to resource constraints and, you know, costs and so forth, I think it's possible, probable, that a lot of small laboratories will stop performing their own LDTs and may send out more samples. And so if, you know, you're a moderate sized laboratory in the middle of Missouri, you know, you validate your tests on your local patient population, but now all of a sudden, if you're getting referred samples from all over the Midwest, that your local population may not be represented in your catchment area anymore. And so that I think that complicates things for the labs that continue to develop LDTs.
Yeah, we had an interesting discussion. If you get a, you know, we do a lot of fish tests, and we get, you know, sometimes slides from outside institutions, and they're just thicker, but the section is mounted, and that just leads to a little bit of an issue with, you know, focusing and stuff. But that's a pre-analytical variable, and the fixation may be different. You know, decalcification may not be compatible with, you know, fluorescent cytohybridization. So that is extremely hard to account for to demonstrate clinical validity, right? You can say, we know that the test doesn't work because they fixed it wrong, or they, you know, but that's the acceptance criteria, right, of accepting a sample. But no, I think it's, so the extrapolation is, of course, that if it's representing or representative for a local population, it may work with the external, but that's a very tough assumption without data, right?
So maybe thinking aloud, you know, maybe this is a good place where some post-market at least surveillance could be beneficial, where, you know, I submit my test with clinical and analytical validity on my local patient population, but then I have some sort of metrics in place to kind of track to make sure that the AVCV that I claimed with the samples I validated with, um, that it's truly representative once I'm running my test, you know, for real with my full patient population, which may involve people from outside my, you know, local region.
I mean, in a way, we're doing that right now. We can't really prevent it, right?
True. Yeah, I don't think this is a new concept for us, but it maybe becomes more important.
I mean, it's basically like the post-market surveillance would be verifying the performance after a while, which could be part of the tech cert or whatever, you know, performance certification or whatever that would be called, right?
Mm-hmm. And we already do a lot, you know, like we do proficiency testing, we've got QC. I mean, we do a lot of things to kind of do that ongoing surveillance of how the assay is performing. So, you know, I don't think this is something we'd have to create from scratch, but, you know, again, it might become even more important if IVCT testing gets consolidated into fewer actors.
So just to clarify, so let's say a lab has, you know, they validate and they do their ABCV for initial submission on a specific, let's say their local patient population, but then they expand the number of tests are coming up from multiple areas. They don't have to resubmit. They would just, this is what we're talking about is the post-market. They would just continue to follow the ABCV with now their increased catchment, right? They don't have to redo their ABCV criteria and resubmit just because they have different samples from different places.
I think that's accurate. We have limited experience with FDA, but we have had conversations with them again where, you know, we are doing validation on a bunch of Minnesotans, and again, primarily white Norwegians. I'm not Norwegian for the record, but there are a lot of them around here. Whereas our intended use is, you know, adult patients. Well, you know, we don't have every type of patient under the sun, right? And so we have gotten a little bit of pushback on that on occasion where, you know, you need to find ways to expand your sample pool for validating your test. And so it's one of those things where, like, can you do that up front, or is it something that you can potentially propose to FDA we will do that in a post-market fashion. I don't know. It's an open question, but I think it's, again, because it is a very hot topic right now, um, it's something that labs will very likely need to be mindful of because I'm sure FDA is mindful of it.
I mean, EGFR is a great example, right? There's clearly different prevalence in different, ancestral groups. And I mean, many NGS assays just by SNP pattern could even delineate the ancestral origin, even if it's, whatever fraction you want to call it. But, typically it's, homozygous or heterozygous either way. You can probably delineate the ancestral state. I mean, with NOMAD or whatever. So that's quantifiable. But, that gets back to this sort of issue, is this, representative of the prevalence in a certain disease. So, I mean, if you would build, I don't know, in the Minnesotan population, that's probably different than in whatever Southeast Asian population. But by genotype, the Minnesotan citizens that have an ancestral relationship with Southeast Asia, that may be actually a higher prevalence. So like subgroup analysis may work. So, but I like I think, Rashni, you mentioned this expansion approach. That sounds like a reasonable approach to say, look, we did as good as we could right now, and it's representative of what we see. But then, after a while, once the test is run a year or two, you can say, look, we did another sort of like a retrospective and figured out what that looks like in other populations.
And I think this really points to, we wouldn't want to, like, for example, write into the legislation, like labs need to do this for every single test, right? So it really demonstrates where every test needs to be analyzed kind of on its own merits. So if I have a biomarker that is evenly distributed in the population, regardless of, gender, ethnicity, whatever, I shouldn't have to do this. But if, there are indications that the biomarker, does have different prevalence in different populations, well, then I need to account for that.
How did you say that?
I mean, the thinking through this though, so I also do germline cancer risk testing. And so you could run into a situation. So for example, we tested every person in Iceland that had colon cancer. And so there's a couple of founder mutations in Icelandic colon cancer Lynch syndrome patients. Would we have to validate or specify this test can be used on Icelandic population, and then are you going to have to expand that to every subpopulation, where, so I think with cancer biomarker testing in terms of treatment, things that are well standardized, EGFR, BRAF, KRAS, etc. I think that's an easier lift because there's a lot of data on the biomarker, and there's all the different categories, right? There's bench research, there's outcome studies, whatever. And so if you can say that, analytically your assay performs similar to what you see in these other methods that are out there, you're probably okay. But, when you start getting to things that there's a lot more variability. So anytime tumor suppressors come in as a biomarker for cancer treatment, do we have to say, okay, our assay is validated for stop mutations in BRCA exons 1 through 20, right? But it's one of those things where I feel like it's a slippery slope where if you're too specific, you're never gonna, like, no company would ever get a claim on anything because there'd be enmities. So,
and I know that CROs usually refer to that as the art of the intended use wording, right?
Yep.
I mean, that really, this is a great example. So I mean, I don't know how to say that, but this is like the art of wording the intended use. I mean, in a way, when we stick to NGS, right, Foundation has given a great blueprint, right? They picked several key cancers, several key alterations, HER2 as the absolute solid tumor, paradigm. They demonstrated that works, thereby showing that clinical validity is present, period. That's it. Like, I mean, it's still a lot of work. So the, other consequences here is, of course, I mean, this fully applies, but so how would you word that for the colorectal cancer assay for Iceland? That's a very interesting sort of consideration.
Well, and I mean, when I've looked at this or thought about this in the past for germline testing, it would be, this assay is intended for identification of mutations in genes X, Y, Z, including, single nucleotide variant, stop gain, blah, 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 and just have it general like that and demonstrate that I can detect all those. So again, it comes down to, is this just an assay? I'm going to put a sample in and get a result out, and that's it? Or is there actually going to be a person and somebody with expertise in there? And, in our experience, having that person in there mitigates the risk. And, kind of coming back to that whole thing, the risk mitigation is really important because, that determines whether or not this would be eligible for a pre-cert, right? And whether or not you could demonstrate I do a good job with this NGS assay, and therefore I can develop more NGS assays without doing, this back and forth with the FDA every single time.
I mean, in the informational sessions that we had with, some of our submissions with the FDA, in those informational sessions you could discuss these points, but that emphasizes the incredible amount of work that would come to the agency, right? Because even with third parties, right, some of these questions are so intricate, I could not imagine that someone can talk immediately on behalf of the agency. And I think even in those informational sessions, very often, at least in my interactions, it's like information gathering so they can go back, discuss, and come up with a solid plan, getting additional input from domain experts. I think the network of experts is a great example, right, where the FDA is already aware that, for some of the other discussion points that we had, like, let's say, an NGS pipeline with AI support, right, that's a huge issue to figure out just technically. But so this point here, I just made that sort of a post-it, but I haven't thought about that before, like, that this is really lab specific, use case specific, intended use case, population specific, right? And then you have to say, all right, basically, like, tailor the validation plan accordingly, right?
Well, it's also, frankly, reviewer specific. Different reviewers will have different levels of expertise and different, frames of reference. And so, you may have one reviewer look at a packet and say, I need more information about your intended use population. And you may have another one look at the same packet and say, your broad intended use statement on your patient population is just fine.
Wow. It's peer review, right?
Yeah, but I think you really nailed it on the head, Joe, where the intended use is the most critical paragraph in your entire submission, because that is what everything is compared against. And at least in my experience in lab, we don't really know how to write intended use as FDA expects it.
No. It's like the analogy that I heard in, talking to so many CROs and experts on this is, in theory, when you talk to a biostatistician, if they take a scientific hypothesis, they know whether it's worded right because it becomes testable. And I think in analogy, most studies, even if, they can be prospective or retrospective, but the hypothesis is usually worded according to the result, not a pre-specified one. And here it's almost like the FDA expects to have a pre-worded intended use that's so specific that everything else can follow from it, including your analytical and validation plan, and you pre-specify that before you even start, right? I think that's probably, I think I read that even somewhere, that the FDA wants to have, basically everything done up front, but I don't know whether that's realistic or actually doable.
Well, that fits with their design control schema, right? That you're gonna develop your metrics, you're gonna test it, and then go down that pathway, and then go back if you need to. Versus, I think what a lot of labs do is, here's kind of what we want to do. We develop the test, and then say, oh, it doesn't work well with indels, therefore we're not gonna, analyze this on indels.
Right.
Right. I think which is a different paradigm. It's not this kind of, here's your outcome that you want, here's the specifications, and if you don't meet it, you go back to the beginning, right? So that's a different thing. Here's another, this is kind of an orthogonal question. So with regards to intended use, how does that fit in with promotional speech, right? So is this gonna be my intended use for my BRAF test is patients with melanoma? Okay, and then now my colleague comes to me and says, hey, I have this thyroid cancer patient. I want to test for BRAF in my thyroid cancer. I think under promotional speech rules, I wouldn't be able to say I can detect BRAF mutation. I can't say I can detect it in thyroid cancer, right? And so you run into this issue of now I'm being treated like. So this is something I talked with some FDA representative with many years ago, and asked him point blank. So in this situation, would I be subject to promotional speech? And they said absolutely. And so that would inhibit my ability to care for my patients because I'm being treated as a manufacturer, commercial entity, whatever you want to say. And so I think that's another angle on this intended use is how much does that hem in my ability to practice medicine?
Well, yeah, and that's just it. Is if we start, tiptoeing into the whole practice of medicine thing, and this is strictly my opinion. I think it comes down to matters of degree. Like if a colleague comes to you and says, I think your test might be helpful for my patient, even though your test isn't intended for that. What do you think? And you talk through as medical professionals one time, that's probably going to be okay. But now if like you start posting on, the internet that you did this and, it worked out great and everyone should really think about it, that turns into marketing and then I'm sure you'd get swatted down by the FDA. But you're right. That's a very gray area. And I know, plenty of people have raised sort of the First Amendment as well as the practice of medicine question related to marketing.
Yeah, and I think if you're out there, tweeting that, my assay branded X is great for this, definitely will get swatted down. But what if I publish a case series? What if I publish case reports,
Yep.
showing that I've used this assay in a way that isn't, I wouldn't necessarily probably put that in my methods. I'm using this off-label, quote unquote, but is that promotional speech now?
Yeah, that's a good question.
To demonstrate CV, right? Because you could use that later on for your expansion, like what we just defined as the expansion approach.
Right, the post-market.
Correct. One other thing that I wasn't quite sure when it comes to this, let's say, marketing authorization. So far, LDTs have been one lab, one territory, no interstate commerce, right? So that's the LDT paradigm, or one of the many factors. So if we have an LDT under VALID, which is an IVCT, is that considered then marketable across state lines?
Yes. It's considered, if it's an approved IVCT, it's considered to be entered into interstate commerce.
So then there couldn't be any more LDTs that are high risk in a way, right?
Well, sorry, go ahead.
If there's, let's say, an LDT, so a lab develops something that's, let's say, considered high risk, whatever that definition is, under VALID that would become an IVCT.
Yes.
Thereby, you're already automatically entering interstate commerce. So you basically, even if you don't want that, right, even if you build the assay to just serve your local, let's say, rheumatology clinic because it's like a first in man use or whatever, and it's considered high risk, you're automatically stepping up the game and being in interstate commerce. You get marketing authorization, so you can, recruit patients from all over or tests from all over the place. But there's no benefit of not wanting that anymore, which I think is one of the benefits of an LDT right now, where you can say, look, we test this locally, we have an expert here who knows what to do with it, etc.
Yep.
I haven't thought about that, but one of the unintended benefits is that you can market, or you would be allowed to market it.
Well, not only that, but you could also sell your protocol to other laboratories now. So today, if I develop an LDT, I can't sell that protocol to other people because then I become an IVD manufacturer, basically. But under VALID, I could sell my protocol to whoever I want because I'm a developer now.
So LDT would basically be an IVCT.
Yes, LDTs go away.
Yeah, so, well, unless it's, just a very simple, low risk
Well, or
No.
it's grandfathered, right? So the grandfathered high risk test will still be in place for a number of years.
Yeah, but LDTs go away. Everything gets converted essentially to an IVCT, and there would be grandfathered IVCTs. But if I develop a test and I only use it in my four walls, it's still an IVCT. It's still been approved for interstate commerce, even though I'm not engaging in interstate commerce. I'm just testing my local population. It's still an IVCT.
Yeah, that's really tricky because the IVCT assume I would sell that, right? The other party would still need to verify its performance,
Correct.
but could also just develop it themselves, but then would have to be an IVCT.
Yeah, so the benefit for the lab that it's being sold to is they could buy an IVCT test protocol and run it as is. They would not be a developer, so they would not be subject to FDA oversight. They'd only be subject to CLIA as lab operations, right? But if they then modify that IVCT, then they do become a developer and would be beholden to FDA and potentially submission.
So, but then if the purchasing lab says, look, we don't have the same baked slide, so we have a different extraction, that's a modification.
Mm-hmm.
So it basically means you either do exactly what's happening right now, or
Mm-hmm.
you immediately turn an IVCT into in now terminology, LDT, thereby you become a developer, meaning an IVCT sold and purchased by another lab would fall under the same modification rules as now an IVD. When you implement it and modify anything, it would automatically turn into an IVCT.
Correct.
Wow.
And so it will create kind of an odd inducement where a laboratory will buy a kit. So this applies to kits too, since IVDs will fall under this. Do I buy a kit that maybe doesn't do exactly what I need it to because I don't want to have to deal with FDA, or do I develop my own internal test, but now I got to submit it to FDA?
Wow.
Well, so just as another wrinkle then, so, thinking about this MAC that Joe was talking about, that's going to require you to publish your validation essentially to get approved, get paid. So if the method is out there, can you just adopt it? It's already been approved. If you don't modify anything, you're not paying a price for it. You're just copying it directly out of a protocol online or in a paper.
I'm guessing laboratories would probably not post their full protocol,
Just like
because there are elements of FDA submissions that are not public. So to prevent someone from essentially swiping your test, I presume laboratories would put that under like IP protections so that someone couldn't do that, I assume.
Yeah, but there's things like the Goal Consortium, right? So part of that is, anyone who's a member of the consortium should have access to the cloud computing infrastructure and pipelines that have been developed, as well as common access to the probes and things like that. So, there are groups that would do that, and I could see, we're academics. We don't necessarily do things that are financially advantageous. That's why we're in academia, right? So I wouldn't have a problem posting all of our stuff, all of our bioinformatics and everything, and just getting it out there.
Yeah, interesting. Yeah, I do have it at 3 o'clock as well. So this has been a really good conversation, everybody. Thank you.
Thanks a lot. And yeah.
Thanks, everybody.
Post the video soon. Thanks a lot. Bye-bye.
Thanks, Joe.
Take care.